And first, I introduce myself. My name is Sammy Tang. I'm a co-founder of Dashu and also the chair, founding member for the KOL series online. We have been conducting this KOL series within the Dashu forum for about 2 years, and it's going very well. Every month, we have great leaders from data analytics, from healthcare, or from technology to discuss the good topics within this area, and it's very well received every time. And we also have our YouTube links from Dashu and in also our website to all the slides, as long as the speakers in agreement, we post them. So most people should be familiar with Dashu, but if you are new, maybe I quickly introduce. Dashu is a nonprofit organization for sure. We're about 5 years of age now of the Dashu organization. All the membership is free. We really open to everyone. We really just hope we can use this forum to exchange ideas, to help promote new research from data science, from statistics, or anything related to data analytics. So we actually, all of us as a full-time working, for example, I'm full-time working in healthcare pharmaceutical companies and the data analytic head. So all of us have a full-time job, but we are volunteers here, try to help the community to grow. And I really appreciate all the good speakers accepted our invitation to also speak for free to all the audiences in Dashu Forum. So we organize international conference and monthly journal club and any technical seminar, including even career development events. So feel free to contact us at dashuinfo@gmail. And here, as I said, that's okay, Bob, I follow your flow. So here, as I said, we need volunteers, either as management or as a volunteer, as student volunteer, or any perspective to help Dashu to grow, to help the community to grow and exchange scientific research. It is very important. And some of the student volunteer put my picture really big over here. Anyway, so thanks for all the volunteers to make the slides and everything to be so vividly to show the faces here for the people contributed so far. And maybe next slide. Yeah, so today's topic actually is very trendy and hot topic. People always talk about AI, machine learning, and how that can be used in the healthcare, system or the drug development. People are wondering about that. Some people say, oh, it's too much a big topic, but actually I do think with the right technology into the right fit for need in the drug development, it will work very nicely. So we were very lucky, invited 2 speakers today to talk about the specific area about AI in drug discovery and repurposing. So our speaker, I'm very honored, actually, Dashu leadership met them in the JP Morgan conference, pre-conferences, and had a very good discussion with both of them. And they are so nice with such a busy schedule they have. They agreed right away to give a presentation at the forum here today, and both of them agreed right away. So really appreciate. I'm going to quickly introduce those speakers, and then I'll give it to the speakers on the floor. So first speaker is Bob. He is the CEO and co-founder of Dr. Evidence, a software technology company that provides automated research to stakeholders across the healthcare ecosystem. And the company answers anything from individual patient questions to large-scale population health topics. Bob has also worked extensively in the public health sector with the Institute for Health Metrics and Evaluation, which maintains the global burden of disease database for the world, basically WHO, and the Bill and Melinda Gates Foundation. Bob works extensively with global academic institutions on their public health initiative. In 2018, that's actually it's a great achievement. He was recognized with an honorary surgeon designation into the Royal College of Physicians of Edinburgh. So he has a very long, very fruitful achievements in his bio. I'm not going to read every pieces, but I really think this is a very distinguished speaker. And our second speaker, also very distinguished, Dr. Mir Sofia Fazeli. She is the president and Managing Director at aditional Outcome Research, based in Vancouver, Canada. He has over a decade of experience working with academic and industry, acting as a principal investigator, project lead, and medical research consultant. His work in evidence synthesis covers a diverse range of methodologies, including systematic and targeted literature reviews of clinical trials and observational studies, meta-analysis, network meta-analysis, and complex statistical modeling studies. So we basically have a MD and also statistic or data analytic-driven combination for today's topic. I think that's a great combination, even in our daily work when we work in the healthcare. So with that being said, maybe I'll give the floor to Bob and Amir, with whoever is the first, and for their presentation. Thank you.
Thank you so much. Let me just.
Sorry, Bob. This is an annotation. I think somebody has left an annotation there. Can you, Sammy? Do you think you can remove this annotation?
Yeah, we'll try to work offline on that, but feel free to continue your discussion.
Okay, Amir, let me just pull up another version. I'm sorry, give me 2 seconds.
No,
I don't know where that's
I don't think this is a version, Bob. If you can click on View Options from above, and then click on Annotation from the Zoom options.
Okay.
View option, annotate, and then click on the recycling bin. There's a bin there which says clear.
Yeah, sorry.
You got
okay.
it.
Yeah,
we're good.
Got it.
Thank you.
Great.
Yeah. Okay. So now let me get back in presenter mode. Thank you. So Sammy, thank you for the intro, and thank you, Dashu, for arranging this. I want to say that you are on the cutting edge of virtual social distancing meetings, which is very apropos for what's going on today. So this is a great meeting. I have my contact screen up, and we'll end with this, but if anyone wants to reach out to Mir or myself, they can do so with any of this information, and we can distribute this afterwards. You can see the slide on our contact info. Yes. Mir, is the agenda slide moved on now?
Yes.
Okay, great. So this will be our agenda today. We will talk about drug repurposing and, quite frankly, novel hypothesis generation. It's not just what they refer to as traditional drug repurposing, but how to have scientific put the inductive work back into drug hypothesis and scientific development, which is heavily applied to drug repurposing. So we'll talk about one of the big themes in the beginning, which is artificial intelligence, a forked road. A little bit of a play on words, but we do have a very decisive road to walk down, and we'll chat about which one to walk down. And then using AI and drug repurposing, we're going to show about a 5-minute video which shows the actual model and some of the techniques used. And the video will stream very nicely over the presentation. And then talk about some of the details of drug repurposing programs that we've applied in a couple of large top 5 pharmaceutical companies on some very dynamic molecules and what it works well on and what it doesn't work well on. And then traditional versus new novel techniques, some of the challenges, and then summary of novel strategies. And then we'll try to get into Q&A towards the back end and leave plenty of room for that. So I'll be presenting the top end, and Mir will be jumping in on a couple of the slides in the middle of the presentation. Okay, so kind of starting out, we're starting with a judgment hypothesis, which is that AI is best used in today's environment in concert of a functional sequencing with human intelligence and technology versus using AI to run total processes, which is, we still think, about 20 or 30 years out, maybe even more. And I'll get into that in some of the other slides. So basically the differentiation between having too much of an expectation on AI processes and what's referred to as narrow AI. So we'll talk a little bit about general versus narrow AI. And in using narrow AI, being very, very specific about what AI is good at today and what it is still very challenged to do. So I wanted to ask some questions, but people can just kind of ping the screens. But I imagine that pretty much everyone nowadays knows the difference between general AI and narrow AI, and I just put some big examples here. General AI is really where something is done from beginning to end. Unfortunately, IBM Watson tried to do this way too early in the process and made too much of a splash in the industry and set a lot of us back, quite frankly. Not picking on IBM, but they're big enough to pick on, where they tried to say, you know, AI is end-to-end drug discovery, a pharmacovigilance solution. Beating Jeopardy was a very specific set of fixed questions. So jumping from the Jeopardy challenge to making decisions by the bench side was too big of a leap, and it set the industry back quite a bit. So that's a good example. And obviously, you know, you can't summon your car to come through neighborhoods and on-ramps and off-ramps and country roads and highways to get to you. But your car today is very good. A good example of narrow AI at self-driving. And it's actually only good at self-driving down certain streets. It's good on self-driving on highways where there's defined lines. And it's building kind of rules along with using deep learning and narrow artificial intelligence. So those are two good examples. So we're going to focus very much on using narrow AI processes for scientific applications of drug hypothesis and repurposing. So just a couple of fun little visuals here along the way. Many of you, some of you might know the reference of this classic iconic image at the top of this hill. This is the famous bone toss in the movie that was done in 1968 called 2001 Space Odyssey. If anyone hasn't watched it, it's classic. But in the beginning of the movie, and then what became famous is the HAL computer, which Hal was trying to answer all kinds of questions. So we're all trying to get to, you know, that revolutionary state. It's quite a journey and adventure. General AI isn't going to make it for us, but narrow AI used along the processes of handing off narrow AI processes to human functions, back to narrow functions, back to human functions is really the applied method here. So how old is average general AI? If you were to kind of, you know, put it in our life years, it's about the age of a 2-year-old baby. If any of you have children, it needs a lot of attention. It can't do anything. It's still learning. It's absorbing. Just like a baby, it needs to be protected, and it needs a lot of inputs, another 5 to 10 years of inputs before it can start being a functioning person. That's really where general AI is on the spectrum. Narrow AI can be a very mature, wise individual, and the narrower the path, the more wise the application of the solution. So again, just in terms of setup, it's important to follow the disciplines of these general rules. Just a couple more setup slides on this. Obviously, average AI still can't discharge a patient from the hospital. And the big reason is judgment heuristics. There's way too many judgment heuristics that need to come into play to do a complex task like that. And this is where we're not going to have the threat of AI running our lives, because people still it's expected that the synapse and just the neural network patterns that happen in the brain, over 50 billion of them, AI can barely do 1 billion of those processes today. So, so we're a far way off from having general AI run big processes. On the other hand, narrow AI is very, very, and now we're going to get starting again some very, very deep specifics. It's great at doing things like pattern recognition, and it's better than doing it than humans. So the classically applied methods right now are companies like Page AI, which are partnered with Memorial Sloan Kettering. One of my board members is on the board of that organization, where they're looking at 30 years of diagnostic images at Memorial Sloan Kettering in New York. And they're huge training sets. So where you have big, large training sets, and we can calibrate AI with deep learning and other methods, neural networking techniques of computing versus programmed computing. That's where you get great benefits. Big word of caution here, though, if there's biases in the data sets, this is a very dangerous method as well. That is that if the definition of, if the way that the radiology images were taken, or the way that they were stored, or the specificity and accuracy and precision of the radiological images over time changed, and now you're comparing earlier images and standard images that have changed, and you're using these incredibly powerful deep learning methods, that's what's called data bias, and you'll end up with a worse result than if you didn't use AI in that case. Even in the narrow AI example, it's super important to note this. If you can't let the system know that there was a point of bias in the data, then the machine will not, it'll give you an aggressive positive answer that will have a huge false negative in it that won't be able to be detected. So super important to realize, even places where AI is incredibly powerful today, if there's biases in the data, we're putting ourselves at great harm and our populations at great harm that rely on those solutions. So using narrow AI with a combination of human knowledge properly applied and an assessment of biases in big training sets that we let loose on narrow AI are the big parts of the discipline. It's very much a journey, as I've said. 2020 is the new 2050, really. We were expecting things to happen by 2020 that now we're saying are going to happen by 2050. So we still have a long way to go. We all still have a lot of work to do. I'm going to run this video now, and you should be able to hear it. Please pay good attention. This is a video where we've applied our AI drug discovery process against the traditional, what they refer to as ad board KOL, so key opinion leader ad board, where we assembled an esteemed group of key opinion leaders. This was for an interleukin 4 and 13 molecule, and then did it again for an interleukin 33 molecule. So this work specifically works incredibly well on dynamic molecules, which are most of the molecules coming down off the bench and into the commercial research pipeline today, where they're immuno-oncological molecules. They can work on GI, they can work on derm, skin, they can work on cancers, all forms of oncology. They can work on immunology. And what we typically do is we put those into prior knowledge key opinion leader panels, and they use their limited knowledge. As you know, key opinion leaders are fabulous, but the big paradigm problem with key opinion leaders is they get into smaller and smaller concentric circles around their knowledge. So the better they get, the more scientifically insular they get about their opinions. There's a lot of research on this where they actually don't see data that conflicts with their cognitive biases, and they rule out data that is good data that doesn't comport and comply with their theories. So we did a side-by-side process for AI with a 10-week AI sprint against about $1 million ad board panel with 6 esteemed panelists for this interleukin molecule. And I'll tell you the punchline after you see the video, but let me run the video here and let me just confirm that it'll run for everybody once it's running here. Okay, Mary, you can hear that?
We can hear it, but we can't see it.
To support the
If you
lifespan
can,
of existing
I think
assets,
you need
several
to
therapeutic
share
areas
your
present high
screen
potential
and
for drug
share the video.
repurposing. But as hundreds of new indications
Sorry.
are explored,
Bob,
the logistics of traditional
pause the video for a
KOL-centric
second?
approaches are not viable to handle this level of.
I will. Sorry, Mary, I'm catching up with you. Two
No worries.
seconds.
If you can reshare your screen again.
No, I'm gonna I have to reshare the let me
YouTube.
reshare the yeah, no problem.
Yeah.
Sorry guys.
Thank you, Mir. You're a very good partner with Bob.
We've been working with each
Is
other.
this all going?
Yes.
Is this
Yeah,
going?
now
Everyone
we
can
can
hear me well so far?
Yes, you got it. So we need
Okay,
to
great.
Valid hypotheses How can we systematically generate clinically valid hypotheses for potential novel indications to support this?
No. All right.
Several therapeutic areas present high potential for drug repurposing, but as hundreds of new indications are explored, the logistics of traditional KOL-centric approaches are not viable to handle this level of complexity. Here, we explore how a new AI process was recently used to identify over 50 novel indications using an innovative hybrid model that combines text mining, real-world evidence, and network-based approaches. To identify relevant indications, several key factors were considered in the clinical framework. We start by exploring the doc search ontology, a robust knowledge graph that helps identify related concepts and their synonyms. We then perform a rapid literature review by applying semantic web, text mining, co-occurrences, and machine learning techniques across a harmonized database of publications, trials, meeting abstracts, as well as drug labels that treat the pathway mechanisms of interest. We then use real-world evidence to explore phenotype similarities and generate hypotheses for these novel indications. The RWE approach in this example draws from a data substrate of 98 million patients in the United States. We can explore and analyze a patient population of interest as we rapidly scan and characterize large amounts of patient data to build robust data engineering and data science capabilities. It would, for example, help us easily search for all adults diagnosed with an immunological condition in their lifetime, constructing a representative patient cohort of approximately 20 million patients. Every patient is then characterized by a rich set of more than 2,500 features, including diagnosed indications, prescriptions, demographic data, lab test results, and medical procedures followed. For one patient, it, for example, would encompass the medical events for the last 7 years. This patient was diagnosed first with a mild form of asthma, was prescribed oral corticosteroids, and afterwards developed a more severe form of asthma and was prescribed a biologic. This level of detailed information for each of the 20 million patients in our cohort gives an incredibly rich understanding of clinically relevant real-world data. We then use an unsupervised learning algorithm looking across those different patient characteristics and across this large number of patients to find patterns. It then tries to group patients with a similar phenotype into different clusters, resulting in homogeneous groups of patients with a similar medical record. One of these clusters, for example, could consist of patients who have infections and allergic conditions of upper respiratory tract. get prescribed a combination of corticosteroids and biologics, or have high neutrophilic blood counts in general. To use this for generating hypotheses with regards to potential novel indications for a particular asset, we define a patient subpopulation for which the asset is known to be effective, the so-called referential. This could, for example, be the subpopulation of patients who are diagnosed with asthma. The assumption is that patients diagnosed with indications for which the asset in question could be effective as well are phenotypically similar to asthma patients and therefore grouped together into clusters. The co-occurrence and clusters between the referential, the asthma patients, and the potential indications, for example, atopic dermatitis or COPD, can thus be seen as a signal of effectiveness for the asset in question. The higher the co-occurrence between referential and specific indications, the more both are phenotypically similar, and the higher the confidence in the hypothesis that this novel indication could also be effectively treated by the existing asset. This co-occurrence and resulting confidence for a particular indication obviously varies depending on the chosen referential representing the patient subpopulation in which it is known the asset is effective. Choosing breast cancer as a referential, therefore, would yield hypotheses for novel indications with completely different confidence levels than when taking asthma as a referential. In summary, exploring phenotype similarities is a robust way to generate hypotheses for potential indications. Confidence in the choice of referential is imperative to get relevant indication results, and indication hypotheses are generated in a systematic and unsupervised way, leading to unexpected, clinically relevant, and unbiased results that can feed into the lifecycle management process of the asset. This case study presents a reliable methodology to reduce development costs, attrition, risk, and time to market.
Okay. I will reshare the presentation. Was
and Bob, here is a,
yes?
Yeah, actually went very well. And actually there are audiences ask questions. The video is very good, very interesting. They were wondering if you can share that later. Maybe you can send a link to us.
Yes.
We can share together
Absolutely.
within the presentation. Okay, thanks.
Yes, I will send the video. So that video was, I'll say a few things about the video. The video was, the software you saw was our doc search software, our AI software that we have. And I'll share the video, and if anyone would like to learn more about it, we can do that.
And I would let the audiences feel free to contact Bob and Amir after the meeting even to ask them questions or collaborations or anything else. They have their contact from the beginning, and maybe we'll refresh that in the end of the presentation too.
Perfect. And you're able to see the presentation again now?
Yeah.
Yes.
Super. Okay. So we'll fly through. I'm just gonna preview a few slides. We'll get through, and then we'll get to down here to questions. So we're hopefully getting a question shortly. Just a time check. Let me check. Okay, we're in good shape regarding time. So a little bit relevant to today's situation, just thought we put in this Edward Jenner story about how, though, just a quick summary, that the AI process I just showed you, the AI process came up with in the life cycle management work in that pharmaceutical company, which is called LCM. The LCM work, the 5-year LCM work, the traditional ad board process came up with 6 indications to pursue for drug labeling, and the AI process came up with 32 viable ones. We reviewed over 750 targeted literature items related to the whole process. We're going to explain in a minute here what the process was. And so 6 versus 2. Now, in fairness to the ad board process, it's not to say that one's good, one's bad, and I'm going to show you I'm going to show you the value of doing 2 together. And this is the big theme here, everybody, guys, is about cooperation It's not one's better than the other, tech's better than provincial knowledge or colloquial knowledge. It's that the two together, we have to work together, just like everything going on in the world right now. We have to work together. And that's the big message on this AI work. So this is just a, Edward Jenning back in the day didn't have the ability to have anything like this. And we're hoping that even with the COVID-19, they're able to do some things with RNA sequencing and synthetic work where they can accelerate the advancement of vaccine. But, you used to have to just, today in the real world evidence case, we can model the effects of data a lot more effectively, and we can find targets without having to try to inoculate, infect, and work with live subjects. So just a good little case study. So this is really what it's all about. The
slide.
Yeah.
It's still showing the previous one. It's not the Edward Jenner slide. Can you turn to?
Are you showing the pyramid slide?
No, it's still the
Okay.
how it works slide. You got it. You got it.
Okay. So this is really what it's all about at a high level. So the pyramid on the left is the classic, tried and true scientific method, 100 years in place, where going back to, the Hippocratic days, you started with human knowledge and expert knowledge, and then it went into a known hypothesis exploration, and then epidemiology is not about proving hypotheses, it's about disproving hypotheses. But by the function of disproving hypotheses, you end up with a then proved hypothesis, essentially, because the pursuit of science is to always disprove. So until it can be displaced. And that's what the traditional method is. What we added to it, and the key thing is added, not replaced. What we added to it is this unsupervised learning approach on the left, where we hit a lot of omics databases to look at pathways. We started looking at drug labels, and then we went over to genomics, proteomics, some other omics databases, mostly genomics. And we looked for those phenotypically similar groups that I mentioned. And we created these clusters, and then we started testing the clusters in real world evidence data sets in the US, taken from Truven, Optum, Humedica, and Anthem, and a couple other places where we had real world data sets. We started testing them for the phenotypically similar groups. And then that's where we came down and did some statistical testing on them, and then cycled them over to the traditional pyramid and running it through then KOL. Now, the KOLs then are able to stand on the taller shoulders of, as they say, of other people, and they could see farther. So that's the method. I'm gonna just go to this one, and I'll go back, Mir, and I'll let you do a few. So in summary, omics databases, the cohort building and looking for relevant pathways, and then moving over to some of the statistical testing in the groups against RWE and the phenotypically similar referential groups that were set up, and then tested those for the potential targets that they might apply for with the patient characteristics and the outcomes in there. So, Mir, would you like to touch on this quickly, or would you like to leave it? Okay,
Perfect.
Go ahead.
Go to the next slide, please, Bob.
This one?
Still not showing.
Do you hear me?
You got it. Okay. So let me first say hi to everyone. Thank you for the opportunity. I want to thank Dashu for the opportunity. I think if there is any good time to have meetings and conferences like this. It would be now, especially in the wake of the COVID-19 virus and infection, which I still believe that we are in the very early stages of the disease, where it hasn't really spread to the parts of the world where the population are extremely vulnerable, and having a multidisciplinary team of computer scientists as well as clinicians and epidemiologists at this point in time can really make a difference in terms of human lives across the world, and then finding vaccination or even a treatment for this particular disease. This particular slide, if you look from the left side, I'll try to move from the left to the right side, because the whole purpose of this meeting and how we can use the AI technology revolves around how we can actually, for this time, especially for this COVID-19 situation, we can use the repurposing, drug repurposing using the AI technology. So the traditional way of finding and approving drug for clinical usage started with identifying therapeutic agents, which took time and obviously money, and it involved a lot of opinion from the key opinion leaders, where they had to gather together the clinicians, and based on their experience, they would recommend that there's a particular therapeutic target that you need to target. And then after that, there was a team of chemists and different people from the pharmaceutical societies, where they work, as well as academia, where they work on the fabrication of potential molecules. At the later stages, the molecule would be tested in vitro, and preclinical studies will be conducted on animal models as well as cell-based assays. And if everything works well, the clinical studies on human beings would start, and you can imagine all of these steps would cost a lot of money and time. And then the trial would go through phase 1, 2, 3. At each of the phases, it depends on whether the study or the drug that have been worked on is actually safe enough to be used by human beings at phase 1. Is it really efficacious, and is it really good to be approved or denied for phase 3? On the other side, in the repurposing part, you can see that there's a signature-based approach and the computer science approaches, where you can bypass a lot of these steps by doing computer analysis and target-based approaches to instead of and inform decisions to the KOLs, and then jump into clinical studies and even bypass phase 1 clinical trials if your molecule has already been approved previously. And this will save a lot of time and energy, as well as financial burden on the innovators. So this would be the model of the repurposing that the AI technology can be used for. I'm going to turn it over to Bob again for the continuation of the slides.
Great, thank you, Mira. And the slide moved on now, Mira?
No, not yet.
Okay. It's, I need to pause and resume.
Now it's moved. Yeah.
Okay. Is it moved to using analytical methods to prioritize indications?
AI-enabled indication opportunity pathway. Using advanced analytics. Yes, you got it.
Okay. It needs to keep resharing. So and we want to get to questions I want to leave the last 15 minutes for questions. I think Sammy would be the best thing, right? So I'll wrap up here in a few minutes.
Yes, thank you.
If you can start assembling questions. So this is just, an overview slide. And again, this is recorded. I'm more than happy to send these slides out too. But it's a summary of the process that we chatted about. The next slide, I'll fly through them, is where we run the unsupervised AI exercise about 3 to 5 weeks. we couple that with the ad board work, and then we generalize, we generate the priority indication list. This could get sped up quite a bit. And then depending if you can run a real world evidence study, you've cut around 4 years off of the study. And if you could run small case studies, you can cut even more than 4 years off the potential time to market. Quite remarkable. This is just some data around the repurposing marketing. We think, I'll end with this, we think that, you know, one of the biggest, the most exciting things going on in healthcare right now is the retooling of the pipeline, commercial pipeline around targeted therapies. And that's been going on for about 10 years, moving from big primary drugs to specialty drugs, and really what will become boutique apothecary drugs and tailored drugs for people's individual, you know, panels and genetic makeup and blood work and things like that. We're not even there yet, but in general, the other big thing from a business point of view in the drug development portfolio, if they can figure out how to repurpose their drugs for other things, that drugs where have already been put $2-3 billion into time to get to market, on average 10 years to get to market, 17-year patent. That's why you see drugs really expensive, because getting new indications is really expensive, and they need to optimize that 7-year of market access pricing. But if you can get better utilization out of those drugs, you want to bring more drugs to market and be able to hopefully reduce the price of drugs. I'll just share one or two more.
So this one shows, I can speak to this one, Bob, from 1960s, which was the golden age of drug approvals by the FDA after thalidomide actually started to show side effects. And so among the side effects, there were also good effects that could be used from that. And you can see that the blue line is the new drug approval by the FDA, as well as the red line is the spending on R&D. And you can see that the productivity gap is quite massive. And the only solution to productivity gap in here would be the drug repurposing, so that you can cut the cost as well as facilitate the FDA approval processes. Next, if you can go quickly, Bob.
Yeah, Miran, let's try to end for questions. These can be distributed.
Okay.
I think.
Actually, I was gonna say, because they're popping out questions already.
Yes.
So
let's have them.
Yeah. Let me talk to the audiences first. You can raise your hand and we try to unmute you, or you can, if you can directly type. We have a bunch of questions typed here. I'll read the first one is, from Peter. Can you talk about, sorry, it's just keep typing so people, can you talk about the business terms if someone were to engage you on an indication targeted project? Also, what is your strategy for overcoming generic substitution for repurposed drugs? And actually, Bob and Mir, you can also click the chat so you can see the questions for yourself.
Okay, great. I can speak to both of those things super quickly. So we work with Mir's organization. He used to be in the Doctor Evidence organization, and he's created his own company, which is the service company that does classic systematic literature review of our organization. But our process is that we do this work over like a 10 or 12-week sprint, and the cost is anywhere from, I'm going to say, I don't know, $50,000 to $100,000 probably per project. It could be a little bit more if it involves a lot of literature, landscape analyses, and some add-on things. Like an example I gave, we did 32 landscape analyses, but we do those pretty efficiently with our tools now. That's US. In terms of the generic, it doesn't really work for generics. The one thing you have to keep in mind is that this process works for more dynamic molecules. The area where it works in generic, and we're doing this with a couple of top companies now, is in safety profiles. So for AEs, so to identify adverse events, because just recently discovered in the last month or two that you can do it for scientific plausibility to figure out where AEs might happen. But generics tend to be pretty monotherapy solutions where they don't have a lot of pathway options, so if that makes sense. So it doesn't work as much in generics, although I would say it'll work in generic if you're in a PV, pharmacovigilance, or in a safety role. It works very well in identifying unknown potential AE profiles. Okay, another question?
That makes sense to me. Another one, does the FDA accept RWE for new indications, or is RWE more for off-label use? Interesting.
Yeah.
Yeah.
Mir, you can jump in on that a little bit.
Yeah, we are also publishing a paper specifically on use of real-world evidence in FDA approvals and how it's been widely used after the 21st Century Act. The paper is going to come out within the next month. It's an extensive work that we've put in to understand. Yes, you can use RWE for regulatory approvals. It depends on really the type of the disease. If it's really rare or orphan disease, you can even win just only by RWE. But if it's really a disease where there are a number of RCTs and previous studies, you might need to supplement the RWE on top of the clinical trials for that.
Yeah, that's a great question and discussion. Actually, we also, I was leading a DIA team. We also have a paper just recently published in the Orphan Rare Disease Medical Journal about using RWE or other external data as a historical control. So yeah, Mir, maybe we can also chat more on that. That's a really good question from Lam on that. So I'll keep reading the questions.
And I can see them now too. Yeah.
Okay. Maybe I'll just read for the audiences. And regarding repurposing, would anyone really commit to the cost of a phase 2 trial for an AI-nominated drug without the traditional preclinical POC studies?
Yeah, Bob, I can take this one as well. So that's a very good question. One of the challenges, and one of the slides at the end that I've put, is about the challenges of drug repurposing in these era. Bob, if you can probably just go a few slides back on 26. One of the problems here is the acceptance of using AI for these proof of concepts as a base of proof of concept studies. The way the approach to do this is the best approach and the most successful approach is to use the drugs that have already been approved. And once you repurpose them, you can get a patent for that. And there are two different types of patents that you can apply for this particular purpose. One set of patent is called the composition of matter patent, or COM patent, which is used usually when you are applying for a new indication. And the second one, which is the one patent that you can use for these repurposing, is called the new method of use, or MOU patent kind of patent. And in that patent, the second one, because the drugs are very informational rich chemicals, there are many different ways to convince the companies to put a couple of $1 million for that, to conduct a proof of concept studies for that. So it is feasible, but you have to go very strong in terms of the rationale. And we are always happy to help also our organizations, Bob and my organization, in sort of consulting and making use cases for that. This is the slide that shows the challenges for drug repurposing.
Thank you. Maybe Bob, feel free to read the question, and the audiences can also hear the questions.
Yeah, sure. And there's, I'm looking real quick at the ones here, so I can dispose of just a few of them.
He's talking about the omics data.
Yeah, the omics. So that was the one I wanted to address. So absolutely, it starts with the omics databases, and predominantly, 70–80% reliance on the genomics data, and that is where a lot of the unsupervised, the unsupervised AI learning work starts with looking at pathways. We particularly look at that through the lens of drug labels, because when you start spidering drug labels that have the same indication, you can look them up. Take Humira's got 13 indications. Any one of the indication for rheumatoid arthritis, I think there's 17 labels with Janssen and all the others. And if you looked at all of those indications and you looked at the pathways they affect, you start to get clues about the multiple pathways and the nuances, and then you move that work over to genomics database, and that's when you start building genomic phenotypically similar clusters, and that's where you can start to do some of the statistical work on the reliability of the phenotypically similar clusters, and to check them as a referential group, if you will. So absolutely. What else? Can you share papers? Yes. So I just got word that the paper that we have written has been submitted to a couple of journals, and it's not yet accepted. So it's in the embargo, but we will share that as soon as it gets we get notice of publication.
Thank you. Thank you so much, Bob and Mir. If we don't, it's getting close to the time anyway. Do you want to share the slides about the concluding slides? I want to share people with your contact information too. I think right now it's black screen.
Bob, can you
Right.
slide where our contact information is? I think it was slide number 2.
Yeah, let me get it. And I just shared in the chat the video link, but I will.
Yeah, and also Peter, want to follow up on the question. If you identify a new indication that is patentable, do you assign full rights to the client? He's very interested for business collaboration, seems like. So
We do. Yeah.
Yeah, it depends on the type of contract, obviously. If absolutely for the work that we provide to our clients, we completely provide that to them so they can take hold of that. But if the person who's asking this question means between the pharmaceutical and a smaller company, that needs to be lawyers that will take care of that. And there are different ways of whether you get royalty for it, or you just sell your patents, or different approaches.
But all this work is done as work for hire for the client. Yeah.
Yes, exactly.
yeah,
We haven't yet started a business where we would just do this and bring it to clients. It's a good idea if anyone's interested in it, in that model, and wants to invest time or money into it. Please contact me.
Exactly. I was going to say, so if Peter or anyone in the audience is like to discuss more specific things or even business collaboration, feel free. This is the contact information. But we do have one audience also have a question. Maybe I'll read the last question. For many immune conditions, asthma and Alzheimer's disease, they may share pathological pathways. Does your method have enough solution to distinguish them resolution?
I think so. I would say that it does, because those are multifactorial. Like Alzheimer's, I've done a lot of work with. There's a great book called The End of Alzheimer's, written by Dr. Dale Bredesen. He's one of the leading researchers. I encourage everybody to read it. He is at UCLA and UCSF. I think he's done some of the most cutting edge work in Alzheimer's, but he identified 32 factors. And you really only need to get control of 6 factors. He talks a lot about the trophic loads on cells. He takes a very novel approach, not just to Alzheimer's, but to multifactorial diseases, which is really the state we're living in now with the existence of a lot of comorbidities. We're no longer living in monotherapy worlds as we're getting more sophisticated. The advancement of science is going to be combo therapies and, multifactorial elements dealing with more than one thing, seeing the patient more holistically and more diversely than just trying to treat one thing. Particularly with immuno-oncology, where you treat one thing and then these 5 things happen, and you have to have a consciousness about that. So in Alzheimer's, I think I'm personally very, very committed to that space for no historical reasons, but I think it's one of the most fascinating spaces going on right now in medicine, and I'm personally miffed at the incidence and prevalence of it, and I think it has a lot more to do with what some of these other therapies are doing, like over lipid management, diabetes, and the effects that it has on the neurological system. So I'm very interested in that, but I do think there's a huge application there. Yes.
Just one quick note here. Bob can repeat the name of the book and the author. If you can write it in the message section, Bob.
Sure, let me do that.
I think Tuju wanted to ask about the atopic dermatitis. I can also complement what Bob talked about, that there's a weighting system that we also use based on the type of the study, the journal, as well as the indication and pathway, to understand which of these indications might be a better indication to go after. The very smart way of doing this, and one of the simple solutions to that, is usually to apply for multiple different indications at the same time rather than just going for one indication. This increases your chance of hitting more number of birds with only one stone. I hope this was a satisfactory answer. That's one of the challenges.
So this is the book up on the screen.
Wow, nice. We can see on Amazon even the price already. So maybe last question. As a company, would Dr. Evidence be submitting patents or doing clinical operations yourself?
No.
Yeah. The patent will be done by patenting lawyers, but we'll do the groundwork for you.
Just because we work for pharmaceutical companies, I mean, we're not in another business. Perhaps we might start that, but no, not in this model.
Okay.
We want to thank everyone so much.
Yeah.
And Sammy, you've been amazing.
Yeah. Thank you. It was really inspiring and a very interactive discussion with you guys even beforehand. So maybe I'll conclude the meeting today. And I really appreciate as honor to have you two both here. You prepare the slides and even the dry run, everything. So I just want to let audiences know every presentation we have here is with a lot of effort behind the scene. And I really also thank all the audiences spend your lunchtime with us every month. And please feel free to contact Bob and me. They are very open-minded. Collaboration should start with people with the right mindset. The same thing here. If you really want to join Dashu as a volunteer, management, anything, this is a fun group. We organize events and everybody learn from each other. Everybody extend their network with each other. So definitely a lot of benefits to be part of the group and also listen to the seminars here. So with that being said, we're perfect on time with only 2 minutes extended. Thank you, everybody. Have a great weekend. Thank you.
Thank you. I just want to extend. I wanted to extend to everyone on behalf of Dashu. If anyone is interested in having a free login to our doc search platform, which was the platform you saw in that video. It's an AI search platform. I would extend it because we extend it to healthcare professionals. They just need to send their name and email. We'll get them registered. They'll get an email from the system with an automatic login, and we won't bother them at all. They just get a system. I think it's the top AI search platform in medicine in the world today. We have 2 of the top 5 pharmaceutical companies using it. So I'll make that, and that's That's a benefit of being a DASHU member. So I will
Member.
make that available to everyone.
Thank you. I'll connect the information. Thank you so much. That's a great benefit.
Thank you.
So
Thank you.
thanks.
Okay.
Have a good one, everyone. Thank you. Bye,
Thank
guys.
you all.
Thank you.
Okay. Bye.
Thank you.
Have a great day, weekend. Be safe.
Bye.